Good morning, students. I welcome you all to third day of our comprehensive lecture series on immunology for CSIR UTC NET examination preparation. Hope you all are doing well. Okay. So today we will be talking about adaptive immune system. We all know the few basic features of adaptive immune system, like this system is antigen specific, unlike the innate immune system. It can recognize diverse repertoire of antigen because it is specific. So specificity and diversity are kind of linked. It exhibits immunologic memory. If it has seen the antigen earlier, next time when it sees it, it will give a heightened immune response. So that's what is meant by immunologic memory. Self and non-self recognition. So it can let us know whether whether it is our own self antigen, that is host antigen, or it is foreign antigen. So we said it has specificity, it has diversity, it has immunologic memory, it can recognize self and non-self, and over the time it can develop, it can mature, and depending upon different exposures, different environment, it can adapt itself. That is why the name adaptive immune system. Now, what are the major parts of the adaptive immune system? Lymphatic system is the main component of adaptive immune system. Now, in the lymphatic system, we have the lymphatic vessels. Okay? The entire lymphatic system has lymphatic vessels. Let me try to draw, if I can, in this limited space. Let's see. So let's assume this is our tissue. And within the tissue, this is our tissue. Within the tissue, we have interstitial fluid. Our tissue is fed by arterioles on one side and venules, let's say, on the other. And these arterioles and venules, when they enter the tissue, they capillarize themselves. Okay, so we have in the tissue a wide network of thin capillaries. Okay. So we have vascular capillaries as well as lymphatic capillaries. So we have vascular capillaries as well as lymphatic capillaries, which are originating because of the capillarization of afferent is the one, afferent and efferent lymph ducts, the one that is taking the lymph out. But what is lymph? You see this interstitial tissue fluid. This is the one, when it enters the lymphatic capillaries, it becomes lymph. So continuous flushing of tissues are occurring due to the movement of lymph through the lymphatic vessels. And since the vascular capillaries are also there, there are immune cells in the vascular capillary which will come out, see the antigens in the tissues. It will mix with the lymphatic fluid, it will see the antigens there, and those intermixing happens at the capillarization. So in between the lymph, let's say this is the lymphatic system of the entire body, and off and on we have this lymphoid organs or lymphoid tissue. So we have the lymphatic vessels connected to the lymphatic lymph node. So we have, this is the afferent, the one that is bringing in the lymph. This is the efferent, the one that is taking out the lymph. And this is our lymph node, let's say. These lymphoid tissues have been given different names in different tissues. We will be discussing all those one by one. So all we need to know is how this lymphatic system is helping the immune system to come in contact with the pathogen. And we saw that it happens mostly in the tissues where the antigens or the blood and the blood cells are coming in direct contact with each other. Particularly in the lymph nodes, because in the lymph nodes there is a lot of capillarization present. The lymph is flowing slowly over there. There is a antigen trapping system present in the lymph node. So most of the antigen and immune cell interactions are occurring in the lymph nodes. So it is very important for us to understand the structure of this lymph node. Now, thankfully this structure is there. I don't have to draw it anymore because it is getting congested over there. Okay. So let's see. This is the lymph node which is being supplied by afferent lymphatic vessels, and the efferent lymphatic vessel is taking the lymph out. It has a cortical region, then a paracortical region, and then the medullary region. Now, within this cortical region, we can see these circular foci-like structures. What are these foci? These foci are primarily made of B cell follicles. So there are multiple B cells. What is B cell? B cell is one of the primary lymphocyte. Okay. And when all these B cells come together, they make foci-like structures which are called as follicles. So here it is the B cell follicle. Now around the B cell follicle, we have T cells. So another type of lymphocytes. And within this, you can see these star-shaped structures. These are the antigen presenting cells. So we need cells which will be presenting the antigens to different follicles. Okay. These antigen presenting cells are majorly follicular dendritic cells and reticular cells. Reticular cells are of two types. One is marginal reticular cells. They are called as marginal because see their position. They are around the margin of the cortical region. These red ones. And the fibroblastic reticular cells. These are the ones, fibroblastic reticular cells. You can compare that from B cells and T cells. If these macrophages or the dendritic cells and reticular cells are much bigger, right? If you compare the size of B cell and T cell with the reticular cell and dendritic cell, you will find these are bigger cells and the number is less. So we have a less number of cells in this region and slightly more number of cells in this region. So this gives a different colored appearance to the lymph node. And that difference in the color has been shown in this figure. So if you see, this figure has demonstrated one green region, which is the outside mantle region. Okay? And the follicles have been shown here. The follicles of this part are being shown here. Since there are more number of cells, we call it dark zone. Now, in this part where we have lesser number of cells, bigger cells, that is mostly the antigen presenting cells are there, that part is called the light zone, basal light zone. And there is one further structure where we have even lesser number of cells and the cells are of even smaller size. Okay, so what is these smaller cells? These are memory cells and plasma cells, which are differentiated B cells. Okay. Their number is much less because they are not actively proliferating. Okay, so they are kept as a storehouse. That is why there is a gradation of color. This is the dark zone, this is the basal light zone, this is the apical zone. And this total structure is called as germinal center. So what is germinal center? Germinal center is present in the lymph node, which is having follicles where B cells are actively proliferating, surrounded by regions where T cells are present, and as well as antigen presenting cells are present. Okay. So this is about the structure of our lymph nodes. Now let's move ahead. So we were talking about the lymphoid organs. Lymph nodes are not the only organs. There are other organs also. Depending upon what role they are playing in maturation of lymphocytes, they have been called as primary lymphoid organs and secondary lymphoid organs. Primary lymphoid organs are basically required for the initial maturation of the lymphocytes. What are those two lymphocytes? We talked about it. B cells and T cells. Okay. So B cells are maturing in the bone marrow and T cells are maturing in the thymus. Now, we may imagine that the B cell name has been taken from the bone marrow, but actually the name has been taken from another structure which is found in birds. It was first observed in chickens. It's called bursa of Fabricius. B cells were first discovered in bursa of Fabricius, and therefore the B is coming from the bursa of Fabricius, okay? Not bone marrow. Although B cells are maturing in bone marrow, but the B cells were first discovered in bursa of Fabricius, that is present in birds, especially in chickens it was discovered. Thymus is a bilobed structure present over our heart, and it is the primary lymphoid organ which helps in the maturation of B cells. Yeah. Now, once the B cells and T cells have matured, they move throughout the lymphatic system and they reach different lymph nodes which are present in different organs. See here I have put the structure of lungs because our nasopharyngeal cavity is directly linked to the trachea and the lungs. Nasopharyngeal cavity is very important from the point of view of interaction with antigens. So as the B cells and T cells are surveilling the entire lymphatic system, they are also surveilling these different organs. And in these different organs, we have sites where there is lymph node like structures. Those are called as secondary lymphoid organs. So those secondary lymphoid organs could be present in your head. So there are multiple lymph nodes which are present in the head, underarms, groin region, neck, chest, abdomen. Okay. As well as there are tonsils and adenoids present in the nasopharyngeal passage. So what we know now that lymph nodes are present at different positions. Nasopharyngeal cavity is one of the primary site where the interaction between antigen and T cells and B cells are occurring. The other secondary lymphoid organs are present in abdomen, near stomach, that is the spleen. Spleen is another secondary lymphoid organ. Peyer's patches is present in intestine. We will be seeing the structure of Peyer's patches. Appendix, which is the extension of your large intestine, it's present here. And the lymphatic vessels, which is present throughout the body. So apart from the lymphatic vessels, all other structures are secondary lymphoid organs, which helps the B cells and T cells to come and interact with the antigens. So we have Peyer's patches in the small intestine, spleen over here, lymphatic system over the entire body. Now, some of these lymphoid organs are very well organized. Some of these are not that well organized. Like lymph nodes is one of the secondary lymphoid organ which has very good organized structure. You can see the dark zone, you can see the light zone, but not all lymphoid tissues could be that very well organized. So we have one group of secondary lymphoid organs which are known as mucosa associated lymphoid tissue. So we call it MALT. So wherever there is a mucosal lining, we have the MALTs present. We know that our entire digestive system, alimentary canal, is lined by mucosal tissue, right? So we have the MALT present over there. So let's see the structure of mucosa associated lymphoid tissue. I'll try to make it let's assume this is our intestinal lumen. And this intestinal lumen is lined by our epithelial cells. Epithelial cells are lined in turn by, what are these? Cilia. We said that cilia propels the microorganism out of the system. Ciliary system is also playing a role in immunity. In between these epithelial cells, we have some larger and flattened cells. Larger and flattened cells. We call these as M cells. Let's say we have one larger and flattened cell over here, M cell. What is the function of M cell? M cell is the one which will allow the antigens present in your intestinal lumen to enter. As you can see, there is no cilia present over here. Cilia is present on the epithelial cells. So if the entire surface is lined by only epithelial cells, the cilia will not allow the antigens to enter the system. If the antigen does not enter the system, that also is a problem. The surveillance system, that is the immune surveillance system, will not be able to encounter those antigens because they are not seeing the antigen. So the immune cells will have to see these antigens. These are the antigens which are present in the intestinal lumen. How do they enter? They enter with the help of M cells. Below this, we have antigen presenting cells, particularly the dendritic cells. So immediately below the M cells, we have our dendritic cells. So what it will do, they will take up the antigens immediately and start processing the antigen, which will then be passed on to the next line of soldiers, immune soldiers, which are sitting again in the form of follicles. What are these cells? B cells. Just like your lymph node, you saw that B cells are present in the form of follicles. Here also the B cells are present in the form of follicles. And around the follicles, we have another group of lymphocytes. What are those? Yes, our T cells. And since this is a tissue, this will obviously be fed by your lymphatic system. So our antigens are coming through the digestive tract, entering with the help of M cells, getting presented by the dendritic cells, and then coming in this lowly organized lymphoid tissue, which is called as MALT or mucosa associated lymphoid tissue. This lymphoid tissue is also rich in one of the particular antibodies, IgA. Okay? We will see later why only IgA, okay, when we will discuss about antibodies in detail. So basically what we are trying to say that this is the organization of MALT, which has a special cell known as MALT cell, which does not have cilia on it and helps in taking up the antigens. Okay. Now let's see the next lymphoid tissue. Bronchus associated lymphoid tissue or BALT. Bronchus. It is the bronchus associated. Bronchi are found where? In lungs. Okay. So unlike mucosa associated lymphoid tissue where it is very well organized, these balls have aggregates of lymphocytes organized into follicles but not very well subsectioned. Like T cells are here clearly marked, B cells have follicles. They have the entire tissue, entire lymphoid tissue in the form of aggregates of lymphocytes. So there you can see intermixing of B cells and T cells. Gut associated lymphoid tissue. Now what is this? Well, this is also gut, right? So GALT, that is gut associated lymphoid tissue, shares morphology with MALT, that is mucosa associated lymphoid tissue. Only thing is, MALT is a broader term because mucosal lining is also present in the respiratory tract, urinary tract. Okay? But gut associated lymphoid tissue, which is having the same kind of structure and is also having M cells, is resembling MALT. Only we can give it again a special name because it is present in intestine. This kind of gut associated lymphoid tissue we call as Peyer's patches. Peyer's patches. So GALT shares morphology with MALT. Both MALT and GALT, that is gut associated lymphoid tissue, have your M cells. They have antigen presenting cells. They have intraepithelial lymphocytes. Intraepithelial lymphocytes are these and are lined by epithelium. IgA is also present in both. Only thing is that gut associated lymphoid tissue is present only in gut, whereas mucosa associated lymphoid tissue is present in all mucosal surfaces. NALT, that is nasopharyngeal associated lymphoid tissue or nasopharynx associated lymphoid tissue, present in throat, nasal passages, especially the tonsils. The difference is these are not encapsulated. These are not encapsulated. So it doesn't have a very well-defined structure like this. And they are without the lymphatic system. Okay, so the lymphatic capillaries are very few or not present. So that is the problem with nasopharynx associated lymphoid tissue. They are lowly organized. However, they have antigen presenting cells. They have B cells and T cells. So the antigens will come in contact with the immune cells, but they will have to depend upon these B cells and T cells to slowly diffuse outside the knoll and then enter the lymphatics. So it will depend upon the diffusion rate, unlike your active lymphatic uptake due to the presence of extensive lymph capillaries. One thing we all should remember here, that lymph system, the entire lymphatic system has no pump. Okay, so there is no active flow in the like the vascular system, that is the blood circulatory system. We have heart to pump blood. We do not have a similar pump for the lymphatic system. When the interstitial fluid is entering the lymphatic capillaries, it is entering through diffusion. So how do we ensure that our lymphatic system is properly surveilling the entire body? For that, we will have to stay active. That is why now in this current situation, when there is a threat of very grave infection like COVID-19, people are emphasizing that we all should exercise, we all should stay active, because the more we stay active, our lymphatic system stays clean, the mobility of our lymphocytes increases, our sinuses stay coagulation free. Okay? So exercise activity is very important to keep your immune system on track. Okay? All right. Let's move on. Now let's talk about the lymphocytes. We have talked about the organs present in the adaptive immune system or playing a role in the adaptive immune system. Now we will be talking about the cells that are important. We learned that B cells are one of the major lymphocytes which are maturing in bone marrow. Okay? So it was first discovered in bursa of Fabricius of this chicken. Yeah, she's so happy. All right, so it's not she. On bone marrow in mammalian cells, obviously bone marrow is helping the B cells to mature in the mammalian cells. Now B cells are the ones which act as very important antigen presenting cells. What are antigen presenting cells? They can take up the antigens just like your macrophages. Okay? Macrophages, what they do? They phagocytose. Similarly, B cells also have the capacity to endocytose the antigen, endocytose the entire or pathogenic organism. And when this entire pathogenic organism enters, they break it down into smaller pieces, and those pieces in the form of, if it is protein, it will be chopped off into smaller peptides, right? So those smaller peptides will then be presented on the surface of the B cell. If it just stays inside the B cell, it is not enough for the other immune cells to see that antigen. Okay, you have taken up the antigen, but now it has to be shown to the, projected to the other immune cells. And that they do with the help of MHC molecule, that is major histocompatibility complex. B cells have particularly MHC class 2. B cells can secrete antibodies, but only when they differentiate into plasma cells. B cells can differentiate into plasma cells, which are called as effector B cells. B cells can also differentiate into memory cells, which we will be talking about later. But when these B cells are not differentiated, these antibodies are present in the form of B cell receptors attached to the membrane. So if this is my B cell, its surface will always have a B cell receptor. These receptors will only help the antigens to be recognized by the B cells. So if there are antigenic particles, they need to interact with the B cell receptors, and then they can enter the B cell, get presented with the help of MHC molecule. So B cells can be differentiated into plasma cells to secrete antibodies, and can differentiate into memory B cells as well. Now let's see about the next lymphocyte. That is our T cells. T cell lymphocytes, they arise in the bone marrow. All cells arise in bone marrow because bone marrow is leading to hematopoiesis. We will see what hematopoiesis is. But they migrate to the thymus by 8th to 9th week of gestation. So in the initial gestation period itself, in the first trimester itself, the lymphocytes have originated in the bone marrow and migrated to the thymus. So T cells are the ones which are helping, thymus is the one which is helping the T cells to mature, which is giving them the proper environment. So this is the structure of our thymus. It has a bilobe structure. As I said, it is present over the heart. Has well-defined envelope, which we call as capsule, cortical region, medullary region, and there are septums. So it's a lobe-like structure, which is partially divided by septa. We call them as interlobular septa. And it is well fed by lymphatic ducts as well as arteries and veins. Okay. So this is the structure of our thymus, which provides a good environment for the maturation of the T cells. T cells we have of different types. One is T helper cell. It helps the other T cells as well as B cells to mature. So these T helper cells, they help both B cells as well as T cells to mature. Mature and activate. Basically, activation. All right. Cytotoxic T cells, which we call as effector T cells. These are effector T cells. What is the immediate effect that they bring? They can cause lysis of infected cells or lysis of pathogenic cells. Pathogen or infected cells. So it causes cytotoxicity. And there is one more class which are known as regulatory cells. So as these cells, cytotoxic and helper cells, are causing the immune system to flare up, there is another set of T cells required which will control this flaring of the immune system, and those are called as regulatory cells. Since they are regulating the immune system, not allowing them to blow too much high. Okay, otherwise it can lead to adverse effects. That is why T regulatory cells are also very important. We will talk about CD4 and CD8 in our later classes in detail. For now, we should know that CDs are cluster of differentiation, that is the cell surface proteins present, cell surface markers present, and based on these markers, the different cell population have been differentiated. Okay, so that they are called as cluster of differentiation. So CD4 is present in T helper cells, CD8 is present in T cytotoxic cells. So B cells and T cells both work together. We saw that B cell maturation activation is dependent upon T cells. So those are two different arms of the immune system, arms of adaptive immune system. Okay. This is B cells contribute mostly to the humoral immune system, because what is humoral? Humoral is the fluid or the vascular system. Just because they are secreting antibodies, B cells are secreting antibodies, and those antibodies are freely available in the soluble form in the vascular system. We call this B lymphocyte mediated adaptive immune system as humoral immune system. T cells, which can never recognize a soluble antigen because they do not secrete anything, they have their receptors bound to their cell surface. Always. T cell receptors are always bound. They will never recognize soluble. Soluble antigens are not recognized. Okay, not recognized. Whereas B cells can recognize both soluble as well as particulate. So that is why T cells is called as cell-mediated immunity, and B cell is called as humoral immunity. Now, as I said, that all these cells are arising during hematopoiesis, and hematopoiesis starts very early in the embryonic stage. You can see that this is the early stage embryo and amnion, umbilical cord. Okay, this is not early stage. I'll say this is the late stage embryo, because the embryo is fully formed, the fetus is almost fully formed. But the hematopoiesis starts in the early stage itself. In the first week itself, in the embryonic yolk sac, hematopoiesis has started. And then by the third month, all these hematopoietic cells, stem cells, they migrate to liver. And by the last semester, they migrate to spleen. So during this development stage, hematopoiesis is occurring in different regions. It starts in the embryonic yolk sac, then in bone marrow, and from bone marrow they migrate to thymus in the earlier first trimester of pregnancy. The T cells migrate to thymus, the other cells migrate to liver, and by the late trimester, they migrate to spleen. So the hematopoiesis has started much earlier during the development. And as we can see, we have hematopoietic stem cells, which will give rise to hematopoietic progenitor cells. And these progenitors will differentiate into different lineages. So we have one lineage where it will develop megakaryocyte, which is the platelet. It is not discussed as a part of the immune system. The other, erythrocyte, which is also not discussed as a part of the immune system. But you can see this part, these cells are actually very important players of adaptive immunity, that is the lymphocytes, B cells, T cells, monocytes, neutrophils, eosinophils, basophils. These are all playing important role in your innate immunity. Neutrophil, eosinophil, basophil, they are granulocytes, whereas monocytes are agranulocytes. So this is how the cells of the immune system are originated. So now I will stop here. We have completed the first part of the syllabus, that is the cells and organs of the immune system. I'll stop sharing the screen. I'll check if there is any chat. If there are any questions from the student side, we can take it up as discussion. No. Okay. So now these will be available. The recording will be made available to you in the Google Classroom itself, as well as I will also be posting a quiz which is related to this topic. So every day I'll be giving you a few questions related to the topics, and then we can, if you have any problem, any discussion, any question, you can also post it in Google Classroom. Okay. Thank you so much. Now I'll end this meeting. Take care of yourself. Take care of your family. Bye.